Hello students, today we are here with our next lecture of human reproduction, and in this video we will discuss about the extra embryonic membranes. Here we will discuss about extra embryonic membranes. In our previous video we had discussed structure and functions of placenta and the classification of placenta on the different bases, that is on the basis of histology, on the basis of chorionic villi, on the basis of behavior during the partition, on the basis of origin. So these are the different types or the basis on which we have discussed the classification of the placenta. In this video we will talk about the extra embryonic membrane. These extra embryonic membranes are developed from trophoblast, or we can say that trophoectoderm. This will give rise to the extra embryonic membrane. These membranes are basically found in the amniotes, like reptiles, birds, and mammals. In these organisms, the extra embryonic membranes are found. Now come to the type of these extra embryonic membrane. What are the types? First membrane is called as Yolk sac. Second membrane is Allantois. Third membrane is Amnion. And the fourth membrane is Chorion. These are the four important extra embryonic membranes. Yolk sac and Allantois, both of these membranes are splenconoplure. Clear? Both of these membranes are developed from splenconoplures, and amnion and chorion are developed from somatoplure. Clear? Splenconoplure means the outer membrane, both of these have two membranes. The outer membrane of Yolk sac and Allantois is mesodermal membrane, and the inner membrane is the ectoderm. Here the outer membrane is mesoderm and the inner membrane is ectoderm. This type of arrangement is called as splenconoplure arrangement. So Yolk sac and Allantois are developed from splenconoplure. In case of amnion and chorion, somatoplure type of arrangement is there. So they are developed from somatoplure. Here the outer membrane in case of chorion is ectoderm. In case of chorion, the outer membrane is ectoderm and inner membrane is mesoderm. And in case of amnion, outer membrane is mesoderm and inner membrane is ectoderm. We will see this arrangement in the diagram also. So here the four types of membrane, that is Yolk sac, Allantois, both are developed from splenconoplure. in which the outer membrane is mesoderm and the inner membrane is ectoderm. We will discuss it later on with the help of a diagram. And amnion and chorion, both of these are develops from somatopleures, in which in case of chorion the outer membrane is ectoderm and the inner membrane is mesoderm, and in case of amnion the outer membrane is mesoderm and the inner membrane is ectoderm. Now come to the diagram. After that we will discuss the function of these membranes. Firstly, we will know about diagrammatic arrangement of these membranes. Suppose this is, I will erase this also. Clear? Suppose this is the embryo. I am drawing the embryo. So, suppose this is the embryo. Clear? So, this I will label as embryo. Here, one membrane is develops from this part, and this membrane is called as yolk sac. This is called as yolk sac. All these membranes are having two layers. Second membrane is develops from here, and this is called as allantois. This is the allantois. The third membrane is here. This is the third membrane which surrounds the embryo. So this is the third membrane. This green colored membrane is the third membrane, and this membrane is called as amnion. Clear? This is the amnionic membrane. So this is the amnion. Now the fourth membrane, that is called as chorion, and this chorion surrounds the complete embryonal apparatus in which the embryon and all of the three membranes are inside. This will surrounds the complete embryo, and here the chorionic villi are there. This membrane is the chorion. All these four membranes are having, this is the embryo, this is the embryo, and the inner one is the embryonal cavity. This is the embryonal cavity. Okay, so this is the embryonal cavity. Here all of the four membranes are made up of two membranes. And here one membrane is also present outside of this yolk sac. One membrane is also there, one outer and the second one is inner. Same one outer membrane is also present in case of allantois. Here the allantois is there, and the one membrane is present outside of this amnion membrane. Clear? This is the outer amnion. Clear? So here the amnion is there. This is amniotic layer. Same as in case of chorion, one more layer is also present inside this amnion. This layer is originate from here, and this is the layer, second layer, which is present inside the outer one. So this is the second layer. Clear? In this way, all of the four layers are having two membranes. Chorion and amnion are the somatocores. somatopleures. These are the somatopleures. These are developed from somatopleures. In case of chorion, the outer membrane is ectoderm. Now we have discussed earlier. The outer membrane is ectoderm in case of chorion, and the inner membrane is mesoderm. In case of amnion, the outer membrane is mesoderm and the inner membrane is ectoderm. Clear? Yolk sac and Allantois, both of these are splenconopleures and both of these are having same type of membrane. The outer one is mesoderm and the outer one is mesoderm and the inner one is ectoderm. Now in both of the membranes, the outer is mesoderm and the inner one is ectoderm. It means this outer one is, the dotted line represents the mesoderm and the complete line represents the ectoderm. Same in case of Allantois. Outer one is mesoderm and inner one is the ectoderm layer. Here the amnion is, this is the amniotic cavity, and this amniotic cavity is filled with a fluid. The fluid is called as amniotic fluid. It is the fluid which provide buoyancy to this fetus. Now it provides the aqueous medium for the fetus for the movement or for the floating. In case of in amniotic fluid, some fetal cells are also present. These are the fetal cells which are present in the amniotic fluid. These fetal cells are helpful in the diagnosis of any defect in the fetus. These are the fetal cells and these fetal cells are used in the technique of amniocentesis. In this amniocentesis, we will diagnose whether the defect is present in embryo or not. If the embryo is defective, then the pregnancy should be eliminated. That is called as medical termination of the pregnancy. So the pregnancy should be eliminated. That's why some fetal cells are used for the diagnosis of defectiveness or the deformity in the fetus. So this is all about the extra embryonic membranes which surrounds the embryo. Clear? Now come to the function of these extra embryonic membranes. We will discuss the function of these membranes one by one. The first membrane that develops is called as yolk sac. The first membrane is yolk sac. So this first one is yolk sac. This yolk sac behaves as embryonal gut. It behaves as embryonic gut or embryonal gut. Clear? This is helpful in the process of digestion. The yolk sac is very poorly developed in case of human beings because all the nourishment comes from the mother's body through placenta for the development of complete fetus. There is no requirement for the much amount of yolk sac. That's why in case of human beings the yolk sac is very poorly developed. But in case of reptiles, birds, this membrane is called as cleidoic embryo. The membranes, the embryos in which the yolk sac is present in sufficient amount, they are called as cleidoic embryos. In case of reptiles and birds, the eggs are cleidoic because they have the sufficient amount of yolk sac. Sufficient amount of yolk sac is there for the complete development of the embryo, because in those animals the egg, after the process of egg laying, the egg is not further in contact with the female body. That's why the complete development is going to take place inside the egg itself. So it has the sufficient amount of yolk sac. But in case of human beings, it is very poorly developed because there is no requirement of this yolk sac. The fetus is going to take up nutrition from the mother's body through placenta. The second membrane is allantois. This allantois, it behaves as embryonal kidney. This behaves as embryonal kidney because it helps in the removal of nitrogenous waste. That's why it behaves as the embryonal kidney, and it is also poorly developed in case of human beings because there is no requirement of this. As the placenta is going to remove its nitrogenous waste through placenta, the fetus is going to excrete or is going to remove out its nitrogenous waste from the body through placenta in the mother's body. That's why it is also very poorly developed in case of human beings. The third membrane is called as amnion. This is having a cavity that is called as amniotic cavity, which is filled with the amniotic fluid, and it is very helpful. The fetal cells which are present in the amniotic fluid are very helpful in the diagnosis of the defective conditions in the embryo, and this will provide aqueous medium. This will provide aqueous medium to the embryo. Clear? So this is the amniotic membrane. The fourth one is chorion. This chorion is well developed in case of human beings because this chorion will give rise to the fetal part of placenta. Fetal part of placenta. That's why it is a developed membrane in case of human beings. It is also behaves as embryonal lungs or the embryonic lungs because it helps in the gaseous exchange. So these are the important functions of these four type of membranes. Clear? So these four are the important membranes present around the embryo, that is yolk sac, allantois, amnion and chorion. Out of these, yolk sac and allantois are poorly developed in case of human beings. This is all about the different extra embryonic membranes. In our next video, we will discuss about the next process that is called as organogenesis. Thank you very much.